Friends, welcome to Gurung's classes. So today we'll study about epistasis. Okay, so let me zoom it a bit. Okay, epistasis, Epistasis. Epistasis. So normally, we have is a pair of chromosomes. Okay, and suppose this is a gene called A, another gene is also which is called A, is present in the another chromosome in the same locus. Or we can make another homologous chromosome. Suppose this is B and small b is also there. So these two are alleles, and then this has a direct impact upon this, or this has an impact upon this. So that means they are affecting each other, but the genes are present in the same locus. Epistasis means how about epistasis means a gene influencing the expression of another gene. A gene influencing the expression of another gene which is present in a different chromosome, and that also in a different locus. This position is known as locus. The site in which the a gene is present is known as locus. So if a gene influences the the working capacity or the expression of another gene which is present in another chromosome in a different locus, then that phenomenon is known as what? Epistasis. For example, there is a gene called G1. There is a gene called G1. And this gene G1 is producing an enzyme, for example E1, and is producing an enzyme called E1. And because of this enzyme E1, a precursor molecule, say suppose P, precursor molecule is changed from P to what? Q. Suppose let me write Q. Gene 1 G1 is producing an enzyme called E1. Because of this enzyme E1, a precursor molecule P is converted to Q, but it is an intermediate it is not the final product. Intermediate molecule. This is just for an example. Q. And then after this, the Q is Q is converted to R, because there is another gene that is called G2, which then produces an enzyme called E2. And the E2 converts Q to R. This R also is an intermediate molecule. Suppose intermediate molecule, and finally this R is converted to S. S is the final product. Suppose final molecule you can write. Fine, or it can be any pigment, or it can be any substance which is produced, and this intermediate R is converted to S with the help excuse me by a gene called G3, which produces an enzyme called E3. And now E3 converts R to S. Fine. So that means, say suppose here we have these three genes working together to get a final product which is called S. Which is called what? S. This is the final product. But here if we if the here, say suppose if the question arises, if say suppose G2 gene remains silent, Okay, if this G2 gene remains silent, then the precursor molecule which has been converted to Q will not be able to convert itself into S. That means the final product S will not be available. That means G2 gene has a direct effect upon G3 gene. So G2 gene is known as the epistatic gene. It has to be present. Okay, and G3 gene is known as hypostatic gene. You can write that also. Hypostatic gene. Okay, so let me give you an example of various types of epistasis. There are various types of epistasis. Okay, like the presence, matters a lot. The presence matters a lot. Okay, for example, let me give you an example of examples of epistasis. Examples of epistasis. Okay, example of epistasis. Let us take an example whereby a sweet pea. Last time we took garden pea, now we are taking sweet pea. Okay, capital L, Lathyrus odoratus. Lathyrus, okay, odoratus is there, odoratus. Okay, so it had two types of flowers. Okay, this one was purple flower and another was white flower. Okay, so for purple, okay, and for white, okay, what was seen? Small p. Make sure when you write small p, small c, okay, be careful. Okay, so when these were crossed, okay, you all know very well, then we get is capital P, okay, small p, capital C, small c. Okay, so all the flowers, okay, they all were purple. They all were purple, so that was why P P C C P P C C. This is in which generation? This is in F1 generation. Okay, now any two plants were taken randomly, okay, and they were crossed, and before crossing, what do they make? They make is what? Gametes. You all know very well. And what matters, what doesn't matter? The number of gametes does not matter, but the type of gametes matter. Okay, so capital P, capital C, capital P, small c, small p, capital C, small p, small c. We can write this capital P, small c, small p, capital C, small p, small c. Okay, so these four types of gametes are formed. So let us make the checkerboard. And while going through this, okay, when you make this in your copy, okay, please make sure you do not hurry, okay, because when you write capital P and small p, you have to be very, very careful, or while writing capital C and small c also. Okay, so we have is four different types of, okay, gametes: capital P, small and small c, again small p, capital C, and small p, small c. Correct? So again here also we have is the same type of. Okay, so now let us P P C C. Okay, or let me write it this way: P P, capital P, small c, capital P, small p, okay, capital C, capital C, capital P, small p, capital P, C. Okay, C, C, P, P, and C, C. Sorry, capital P and small p, capital C, small c, capital P, small p, small c, small c. Okay, capital P, small p, capital C, capital C, capital P. Okay, small p, capital C, small c. Okay, then small p, small p, capital C, capital C. Okay, small p, small p, capital C, capital C. Okay, okay, and what we have is capital P, small p, capital C, small c, capital P, small p, small c, small c, small p, small p, capital C, small c, and small p, small p, small c, small c. Okay, so after getting this 16 different types of combination from here, what we can write is, okay, now this is what? PPCC, this is purple. Okay, so 1, let us cross this. Okay, now this, okay, this was also found as purple, so purple. Okay, this P and C together was there, this was also found to be purple. This P and C was also there together. Okay, so this was found to be purple. Okay, this was also found to be purple. 5. Okay, but this, okay, when capital PP was there, okay, but only there was no capital C, not a single capital C was here, so this was supposed to be white. Okay, this was supposed to be white. Okay, so white, this purple. Okay, this again white. Okay, this purple, this purple. Okay, 1 2 3 4 5 6 7, 1 2 3 4 5 6 7. Now this is also purple. Okay, purple. Now this white, this is white. Okay, this purple. Now this white, this is white, and this is white. So 1 2 3 4 5 6 7 8 9. 1 2 3 4 5 6 7. Okay, so the ratio between purple is to white is 9:7. Okay, so from here what we can take out is, for the purple color, okay, if you want to see the purple color, okay, then capital P itself is not capable of giving the purple color. Okay, the capital C also has to be there. No matter if one capital C is also there, also we can have is a purple color. Okay, but if only P is there, okay, if only P is there, then it is not able to give purple color. Okay, so here what we can say is, this is a direct effect of one gene upon the another gene. Maybe the gene could be in a different locus, but the presence of capital C only, okay, makes capital P capable of giving purple color. So this is known as what? Epistasis. And this type of epistasis is known as, we can write here also, this type of epistasis is known as complementary gene. Complementary genes. Okay, so the question might come, complementary genes example, then you can give this example. So thank you very much.